This is a special episode of the Immunology Podcast, Immunology 2025, Day 1. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology Podcast, where we have conversations with immunologists. As you all know, today launched the start of Immunology 2025, the annual meeting of the American Association of Immunologists in Honolulu, Hawaii, and Brenda, myself, and the Immunology Podcast team are attending. Drop by the Immunology Podcast booth in the exhibit hall to play some games, win a gift basket, and find out how you could be featured on an episode of the podcast. Today and every day throughout the meeting, we will be releasing special episodes discussing our favorite sessions of the previous day. So if you weren't able to attend, we've got you covered. We're also very excited to be hosting a live show of the podcast on May 5th. Join us in rooms 317A and B at 10 a.m. for a conversation on trends and perspectives in immunology, drawing from our 4 years of candid conversations with researchers in the field. We're going to kick off things in just a minute, but before we get to that,
if you're in Hawaii, we invite you to join STEMCELL's networking social event on Monday, May 5th, starting at 7 p.m. We'll be at the Hideout at the Lalo Hotel, located at 2299 Kuhio Avenue. See you there.
all right, Brenda, what do you think of Hawaii?
Ooh, aloha, Jason. It's really nice. It's really nice. I have to say, I like the sunshine. Sometimes it's good. And this, the sea and the beach. It's been nice. I'm still jet lagged, but that's okay.
You're still jet lagged?
I'm telling you, I'm waking up so early. Also because our neighbors were so loud, so that didn't help. But it's fine.
Yeah, I think I woke up at 3 a.m. this morning and then stayed up till 5 and then went back to sleep.
Fun.
Till 6:30.
Yeah, that does not sound, at your age, that's not advisable.
No,
Jason.
but I went to bed at 9 p.m. like an
I
old
see.
man, so that was good.
Well, we'll survive. Anyway, the sessions start really early. Not today, but tomorrow we have to get ready. 7 a.m. start. All right, Jason. So we had a few sessions already today that we attended. It was really nice. I think it was a great start. I have a couple of cool stories to share, but gentlemen first.
Oh, wow.
You want to share?
Really? All right. Okay, so I'll hit you up. So my first set, I went to 2 sessions, right? It's kind of a smaller set today. First one was adaptive immune responses to microbial infections.
You're right up your alley.
Well, kind of. It's not innate immune responses, and it's not the epithelium or the microbiome. So it's a little adjacent. I'm broadening.
All right. Out of your comfort zone. I like that.
So the first paper, or first talk, I guess, is about B cells. It is B cells produce anti-inflammatory cytokine IL-27 to reduce inflammatory murine colitis with Shai Wu, who's a postdoc at UT Health San Antonio. So high level, B cells do something cool here. And they found that IL-27 is induced in B cell follicles during experimental models of colitis, this DSS model, which is an injury model. And if you knock it out, the mice get worse. So they're finding that IL-27 is protective against this model. And then they tied that to IL-27 is responsible for producing IL-22 secreting ILC3s.
So what's the ILCs? What's up with them?
It's always ILCs. And ILC3s with this IL-22. IL-22 is wound protective. So in DSS, IL-2 drives STAT3, which protects the gut. Pretty classical pathway. And they also find that it promotes IgA production, which also then binds some of the toxins and some of the harmful bacteria. And so if you lose this with a knockout, the colitis gets worse. And they were also able to show that it's induced in the follicles of IBD patients. So again, somewhat preliminary work, but cool. Finding some B cell mechanisms for IBDs, always fun for me. The next one is colonocytes targeted by Citrobacter rodentium elicit epithelial MHC2 restricted help from CD4 cells. It's by C, stands for Christopher, but he goes by his middle name, Garrett Wilson from the University of Alabama, Birmingham, Hirsch School of Medicine. So just for people to know, Citrobacter rodentium is kind of an analog of E. coli in mice that's like invasive E. coli. So it's really used as a model of, food poisoning or just E. coli pathogenesis in mice compared to humans. That's because mice don't respond to E. coli the same way. So mice lacking MHC2 have greater bacterial burden, which is already known. But they found in this case that MHC2 is required for the mucosal retention of specific T helper cells and that it prolonged that IL-22 STAT3 pathway. And what they really found that was interesting is that this is true even after the bacteria are gone. So Citrobacter rodentium clears out in a specific amount of time, about 28 days. And they find that this effect, and they use timed induced knockouts, that this effect occurs even after they're gone. So these memory cells, which are memory against Citrobacter, these T4, CD4 positive memory cells, they are kept in the gut by colonic MHC2, right? Because remember, epithelial cells express MHC2.
Oh my! So this is MHC2 in the on the epithelium.
Epithelial cells.
Okay.
That keeps the memory cells present.
Oh.
And holds them there even after the infection is cleared.
But is this MHC2 class expression continued after the infection?
Normally, yes.
Yes.
It's like epithelial cells are immune cells. Gotcha.
Damn you. Damn you. All right. You're making a good case.
And last one from this section is maternal-derived anti-commensal IgG promotes the induction of colonic peripheral regulatory T cells.
Ooh.
From Zhongshuan Zhu, the University of Alabama, Birmingham as well. So, mice and humans, you get some IgG from, sucking on the mommy mammary glands and get all that good IgG in your gut. And as a reminder, that FcRn receptor is how that gets absorbed from the lumen into the epithelium and then back into the blood and everywhere else, right? And circulates. So they found, and there's this other ROR gamma T positive, FOXP3 positive Treg called a pTreg, which is important for intestinal homeostasis. And they differentiate between 15 and 20 days of life. So now, having told you all this, the first finding they have is that if you knock out epithelial FCN, so if you just have an FcRn knockout in general, there's less IgG in the mucosa. IgA tends to be in the lumen. IgG is that mucosa, the mucus surface layer of the epithelium, right on that border of the cell. If you knock it out, there is no IgG in the mucus. So it's important for transport. They do a tissue-specific knockout and establish that it's the epithelial receptor that matters for the FcN receptor. But you also get less pTregs in this case. And so what they find is the development-dependent antigen exposure through what's called this goblet cell-associated antigen pathway. So, right, you have these antibodies, they're coming in. There's all these IgGs from mommy, which are somewhat also against self, right? Because your mommy has antibodies against the baby.
Oh.
Right?
Okay.
And so that's part of how you train. The baby trains what it is these antibodies. They're getting, and they bind, right? To these antibodies are going to start binding to baby antigen because you already have this in the circulation, right? Because mommy's antigenically exposed to babies, but now it's feeding the baby these IgG.
But are so like daddy babies antigens? It's like antigens that are not expressed on the mother but are expressed on the baby.
Right, exactly.
Okay.
Or recombination, don't forget that, right? So then that's passed on, right? Because these are anti-commensal IgGs. That's passed on. So now your baby's eating those with the antigen attached to the antibody, and that gets absorbed through the goblet cells through this gap pathway. But that antibody, that FcN receptor on the epithelial has to be there, right? If it's not there, you can't transport it. If you transport it, then you get these pTregs that help develop host identification of the baby. So the baby knows from its mother's antibodies what it is.
So this against babies' antigens or against commensal antigens that the baby's also gonna have?
The commensal antigens the baby has, which include but it's the mom's IgG.
Yeah.
Against the baby's commensal antigens because they're in homeostasis.
Yeah. That's cool. Okay.
After the baby's born, when these develop, you have to have this IgG pass through to train. So the baby's Treg repertoire is being trained by the maternal IgG against anti-commensal.
Got it. Okay, that makes sense. Wow, that's cool.
Yeah.
All right.
So that was wild.
Yeah.
All right. And then in the afternoon, I did immune therapy in cancer. And I'm going to talk about a couple of them. The first one is targeting RAGE-mediated immunosuppression to enhance radiotherapy efficacy in glioblastoma. Saidi Soleimanian from Oklahoma University Health Science Center. So this talk went a little fast. I don't really know what RAGE ligands are other than they're part of signaling. But they're enhanced by radiation in glioblastoma patients, which is a type of brain cancer. So glioblastoma, you get chemo, then you get radiation. Or they'll cut it out and you get radiation. But they found that inhibition of RAGE enhanced survival in mouse models. and that you could target RAGE reduces the presence of G and M MDSC. So it's removing these suppressive macrophages. And so now there's a phase 2 study with a RAGE inhibitor, the same one they use in the mice, ongoing, of which they didn't really talk about the results. But that's cool to see. The next one I thought was super cool. This is the potential for p53 reactivators to increase the sensitivity of aggressive gynecologic cancers to immunotherapy with Lane Smith at the University of Mexico. This is preliminary work, but there's a set of curcumin analogs from turmeric, right? Curcumin's that active ingredient, that are analogs that bind to p53. And what they do is they override p53 mutations and make the mutated p53 that's an oncogene function normally again.
Oh.
So one analog, which is HO3867, reinstates the natural wild type p53 transcriptional program. And as a result, they see the PD-L1 re-regulate normally. And so they're wondering if it can be used as an enhancer to immunotherapy, because you can't overcome this p53 mutations, right? Like are very common cancers we all know. But this is going, yo, you're mutated. Here's a drug that makes you behave normally.
All right, because can we just remind when p53 is mutated? So p53 is important for
cancer everywhere.
No, okay, but what is the normal function of p53?
It causes you to kill yourself in the situation of DNA damage.
Exactly, right? And
it self-inhibits.
But this is so the most common mutation of p53 is this one that prevents the proper the trimerization of the...
Yeah, and so that you don't kill yourself when it senses DNA damage.
Okay.
Right, it turns off the apoptotic signal.
Okay, and then with this compound,
the transcriptional program you see for a mutated p53 reverts back to wild type p53.
Okay.
It binds p53 and restores its function.
Yeah, maybe it restores the tetramerization.
It's some type of bridge or something.
Yeah.
They didn't there's no crystal structure, none of the things they want as a follow-up. But it's an interesting concept for drugging something.
Yeah. Huh.
Yeah. All right, what'd you get, Brenda?
Okay, I basically had an autoimmune morning sessions. So I'm just gonna...
Did you need some prednisone?
Yeah, no, not yet, but almost. So there's a couple of really cool stories that I want to share. The first story of the morning was by Zachary Shaheen. The talk was entitled "Early Exposure to Frequent Microbial Infections Prevents Both Spontaneous and Immune Checkpoint Inhibitor-Induced Autoimmune Diabetes." Oh, that's a mouthful. So he's from the University of Minnesota. So there were a few people from Minnesota. And I think basically it's really nice because we know that autoimmune diabetes can be a side effect of checkpoint inhibition, and also people develop spontaneously autoimmune diabetes, type 1 diabetes, and they studied in the context of not mice that develop diabetes spontaneously, and they show that although not mice develop diabetes in a specific pathogen-free environment, so that's like the normal way of having a mouse. If you have a kind of a dirty, quote-unquote, dirty animal model, which they call normalized microbial experience, NME, that actually protects the mice from developing diabetes. And I think it's also upon PD-L1 blockade. And what?
Are you doing a microbiome review right now?
Kind of, I guess, yeah. So basically, they don't show... They show basically that exposure to microbes in early life, that there's like an appropriate time frame for this, can protect the mice from developing diabetes as a consequence of checkpoint inhibition and also as kind of normal development. And basically that's kind of the general idea of this talk. I think that's really cool because it gives us another understanding of, on the one hand, like how mouse models really differ from real life, I guess. We don't have specific pathogen-free people walking around. And the importance of the immune modulation by the microbiome and this mice. So it's really cool story. So after that, it was Luz Mariel Medina Sanchez from the University of Pittsburgh. And her talk was called "Unlocking the Secrets of T. Arnold a commensal protist that restrains virus-mediated loss of oral tolerance to gluten. And I also thought it was such a cool story. Basically, the question is, you have this increased incidence of celiac disease in recent history. So what are the mechanisms behind this? Why do people get celiac disease to start with? And basically, they study the idea of oral tolerance and how when you have celiac disease, you're basically lost this tolerance. And they zero down on a gut commensal protist that they find, and apparently they sequence it. They find it in the mice, and they sequence it. Apparently, it was not known. They called it so it's a Trichomonas arnold. So that is this new this new commensal protist they find, and they kind of show that it can very interestingly can modulate immune responses and loss of tolerance if mice are being colonized by this particular microorganism. This correlates with things like Treg development. And if they infect mice with reoviruses, which can cause loss of oral tolerance, they have this OT1, OT2 OVA model. And the presence of T. Arnold can prevent this loss of tolerance. And they show that they do this with OT1, OT2 cell transfer, and they immunize the mice with OVA peptide, the target antigen, and they also show that T. Arnold can influence DC polarization towards like less costimulatory molecule expression, less IL-12 production, and this is through IRF1 expression in this disease, basically, it's not clear how they do it, so they're studying that better, but they do see this influence on DC phenotype. And so they do all this initial work on an OT model and OVA, but they also end up moving into more of what they're actually interested in, which is celiac disease and recognition of gluten peptides. And they use tetramers from Mark Davis's lab to pick up gluten-specific T cells. So they pick one of the peptides from gluten, and they look into gluten-specific T cells in these mice, and this allows them to see, CD4 cells as well, Tregs, and they find that after this virus infection, basically this results in a upregulation, an increase in the number of Th1 cells that are specific to this gluten peptide, and this happens if in kind of mice that don't have this T. Arnold, but if they inoculate the mice with T. Arnold with this protist, this results in kind of expansion of Tregs and prevents the development of celiac disease in these mice, and there's like gluten response.
You're 2 for 2 for microbiome.
Yeah, that's true. Microbiome is everywhere, but in the context of autoimmunity, we are so much influenced by it. So I think it was really cool. Really picked up some things from my own research here. And then we had another cool story was from Gonzalo Nogueira from the Institut Pasteur from Paris. And I had to say it was a bit of a tough talk to follow, but I thought it was really interesting in the sense that this talks about early thymocyte development, and I think that the interesting so the talk was titled "Embryonic Thymocyte Epithelial Cell Crosstalk Plays an Essential Role in the Establishment of Adult T Cell Tolerance." And basically what he saw is that different times in development, there's different waves of thymocytes that go to the thymus, so from the bone marrow to the thymus, and then you have development of T cells of different types. And the thing is that if you inject anti-IL-7 receptor alpha antibody into pregnant females, you can interrupt the very early waves of this thymocyte into the thymus, kind of this early waves of T cells being developed in the thymus. And this is what he shows is that, of course, you get less T cells out, but the interesting part is that this the absence of these cells at this particular time in development also seems to have some long-lasting effects on the structure of the thymus itself. So it's kind of a two-way street. The thymus is training the T cells or the thymocytes that come into it, but on the other hand, the presence of these cells also will affect the structure of the thymus itself. And so basically it shows that if you reduce this first wave, this has reduced, affects the structure, reduced number of mature medullary epithelial cells. You have reduced output of tolerogenic cells, of FOXP3 cells. And basically this, in the end, has effects on like subsequent waves of thymus-seeding progenitors of the T cells, that the thymocytes that go into the thymus, and then they will become the actual T cells in the periphery. So this has kind of long-term impact into the maturation of the thymus epithelial cells, and therefore in future output of the thymus itself. And so I thought that was really, really interesting. And then another story that I liked, I think, was from Catherine Block from the Jameson lab, the lab of Stephen Jameson, also University of Minnesota. We had him in the podcast recently. And she studied CD8 T cells in vitiligo. Vitiligo is this disease, what happens in which you have mostly CD8 T cells that are attacking melanocytes on your skin, and therefore eliminating them and eliminating the color in your skin. So basically you end up having patches of white skin. And this is a fairly common side effect of checkpoint inhibition, particularly for melanoma patients. And actually, I think if I remember correctly, it correlates with better prognosis of the result. That you have very active cells that not only attack your melanoma that expresses certain proteins like melan A or MART1, but they will also affect your actual skin cells that have the same proteins. And what it was pretty cool is that they took a melanocyte antigen, a different one, not melan A, but TRP2. And they showed that basically they had these mice that they were knocked out in TRP2. And this didn't really affect anything externally in the mice, but basically that antigen was gone. And it was cool because they compared the idea was to compare the T cell repertoires between the mice that are knocked out and the mice that actually do have TRP1 and therefore would be presenting TRP1 in the thymus. And in order to have so one of the issues with finding antigen-specific T cells is that they're very rare in general. So that's not that easy. So if you have an ongoing immune response, sure, but if you don't have an ongoing immune response, you're not going to find those cells, or it's going to be really hard. So what I think is cool is that they first had mice that had vitiligo, that they have active recognition of this protein. I think they vaccinated for this protein. And then they found, from this very active disease, they took the CD8 cells that were recognizing this peptide, and they sequenced their TCRs. And then what they did, they just chose two TCRs. One of them was expressed only in mice that were knocked out, and none of them were expressed in wild-type mice. And then they had another TCR that was kind of fairly expressed similarly both in wild-type mice and knockout mice. So the idea is that one TCR is not present if you have thymus expression of the antigen. Therefore, this indicates clonal deletion. And then you have another one that is there present regardless of whether there is presentation of this peptide on the thymus, within the thymus development. And so what they did also is they took the V-beta sequences from these TCRs, because the beta chain is the one that gives most of the specificity of the most variable, the CDR3 domain. It's located in the beta chain. So it's most of the recognition of a TCR will come from the beta chain. And so they made retrogenic mice expressing that beta chain, and then whatever alpha chain the T cell expressed. And what they did by doing that, they really enriched in TCRs specific for this TRP2 antigen. And therefore, it allows them to study more T cells, more antigen-specific T cells, in a context in which there was no inflammation, there was no like clonal expansion. So this is a really cool system. So if you're studying T cell repertoires, this is a good way to go. What do they see? So they use the beta chains from these two different TCRs, and they show again that they only saw them really developing. So the TCRs that were derived from the kind of this one, the TCR that was not present in wild type mice, if they looked into the binding to tetramers, for example, that was a stronger binding, indicating higher avidity. And in the mice, wild type mice, this higher avidity was not present. So only those TCRs with lower avidity survived the thymus. So basically, they show that this really proves, or it really strongly indicates that in mice that are expressing the self-antigen, there will be a certain degree of clonal deletion of high avidity TCRs, but not everything. And this is really a very, I think it's a really good way of showing this clonal deletion. But it's not. But the important thing is that it is not complete. So definitely, that is the kind of the different degrees of avidity are reflected in this deletion. So I think that was really cool. And another just last story from Nicole Wilkinson at UCLA. She studied the very important EXIST complex and its role in kind of autoimmunity. So EXIST is this complex that keeps the second X chromosome in females. What's the word? Inhibited or
silenced.
That's the word. And so basically she talks about what if you knock out this EXIST, if you prevent the formation of this complex and you have double expression, she showed, which are the mechanisms by which the presence of this complex alters the T cell effector function. And this is important because women have a much higher incidence of autoimmune disease. And it's unclear, sometimes it's hard to tell if it's a hormonal situation. We do know that hormones play a big role, but also the fact that you have this not permanent, because EXIST, we thought it was kind of a very strong inhibition, and it's not, and it does seem to be activated in T cells, and this might be correlated with this increased propensity to autoimmunity in females. So autoimmunity all over.
It's important.
Yes, indeed.
Microbiome helps stop it.
Yeah.
All right. Yeah. Well, that brings us to the end of our first Immunology 2025 episode. Don't forget to follow us on X at ImmunoPodcast or Blue Sky at ImmunologyPodcast.com. Find out what we're up to at the meeting and visit us at the Immunology Podcast booth on the exhibitor floor where you can win a gift basket. Check back here tomorrow for another episode recapping day 2 of the meeting. See you then.